Welcome to today's lecture on Plant Biotechnology, Unit 1, which is extremely important for CUET PG 2026 as well as for your conceptual foundation in plant sciences. In this unit, we will be dealing with how plants can grow artificially in laboratory conditions, how single cells can regenerate whole plants, how valuable plant germplasm can be preserved for decades, and how elite crop varieties are multiplied commercially using tissue culture. Please remember, this is not only an applied topic but also a high weightage scoring area in competitive exams. CUET frequently asks definition based MCQs, matching type questions, sequence based questions, and principle oriented conceptual items from tissue culture and micropropagation. Therefore, as we move slide by slide, I will continuously highlight the points which are exam oriented. Let's begin. Before going into details, let us see what exactly this unit covers. We begin with the introduction to plant biotechnology and the concept of totipotency, which is the most fundamental biological principle behind all tissue culture techniques. Then we will discuss cryogenic preservation, popularly called cryopreservation, which deals with ultra low temperature storage of plant materials. After that, we move to organogenic differentiation and the various types of in vitro culture like seed culture, embryo culture, callus culture, organ culture, cell suspension culture, and protoplast culture. Next, we explore micropropagation and its methods such as axillary bud, Meristem culture and shoot culture. Finally, we will compare organogenesis and somatic embryogenesis and analyze the advantages and disadvantages of micropropagation. For CUET PG, such outlines themselves are useful because sometimes questions comes like which of the following topics are included under micropropagation or which technique is related to germplasm conservation. What is plant biotechnology? Let us first understand the meaning of plant biotechnology. Plant biotechnology can be defined as the application of biological techniques, particularly tissue culture and genetic manipulation, to improve plants for agriculture, industry, and scientific research. Here, three things are crucial. First, we are using laboratory based biological techniques. Second, the major tools include tissue culture and genetic engineering. And third, the purpose is improvement either in yield, quality, disease resistance, stress tolerance, or metabolic production. The key features include growing plant cells, tissues, or organs in vitro, which literally means in glass vessels rather than soil. These cultures are maintained under strictly controlled sterile conditions so that bacteria or fungi do not contaminate the culture. An artificial nutrient medium replaces soil and provides minerals, salts, sugars, and vitamins. Finally, plant growth regulators such as auxin and cytokinins control differentiation and morphogenesis. For CUET PG, remember this definition clearly. Many MCQs simply test whether you know the plant biotechnology primarily involves in vitro culture and gene manipulation. Importance of plant biotechnology. Why is plant biotechnology considered so important in modern agriculture and conservation biology? One major application is the rapid multiplication of elite plant varieties. Suppose a farmer or breeder develops a superior plant with high yield or disease resistance by tissue culture. Thousands of genetically identified copies can be produced within a short time. Another important role is the production of disease-free plants, viruses, especially in vegetatively propagated crops like potato. sugarcane, and banana cause huge yield losses. Meristem culture is commonly used to eliminate viral infection. Plant biotechnology is also crucial for the conservation of endangered plant species, where only a few individuals remain in nature. In addition, it is used for genetic improvement through transformation, production of valuable secondary metabolites like alkaloids or flavonoids, long-term storage of germplasm, and overall crop improvement programs. For CUET PG, remember, secondary metabolic production is often associated with cell suspension culture, and germplasm preservation is linked to cryopreservation. This slide represents the core principle of plant tissue culture, totipotency. Totipotency refers to the ability of a single differentiated plant cell to regenerate into a complete plant when provided with appropriate nutrients, hormones, and environmental conditions. Plants naturally retain their developmental flexibility much more than animals. In animals, differentiated cells usually lose the ability to form a whole organism, but plant cells can often revert to a meristematic state and then redifferentiation. The implications of totipotency are immense. It forms the basics of cloning plants, regenerating genetically transformed cells, and develop complete plants from callus or single protoplast. For CUET-PG, this is a very common question. Who proposed totipotency? Or which principle forms the basic of plant tissue culture? Answer in the comment section. In-vitro culture literally means growing biological material in glass containers rather than in natural soil. In plant biotechnology, it refers to cultivating plant pods outside the parent plant on an artificial nutrient medium under aseptic conditions and carefully controlled physical parameters. These conditions include temperature, usually around 25 degrees Celsius, light remains such as 18 hours of illumination and 8 hours of darkness, and appropriate humidity levels. Because of the system is artificially and isolated from nature, every factor from minerals to hormones must be supplied externally. This allows scientists to precisely control development and study plant physiology in the simplest environment. CUET-PG sometimes tests the meaning of in vitro or asks which factors are controlled in tissue culture. Remember, microbes are excluded and environmental parameters are regulated. General requirements for tissue culture. To establish any tissue culture experiment, several essential components are required. First is the explant, which is the plant part taken for culture. It could be a leaf, stem segment, bud, or root tip. Next is sterilization, because contamination by bacteria or fungi is the most serious problem in tissue culture. Surface sterilants such as sodium hypochlorite or mercury chloride are commonly used. A nutrient medium such as morassic and soot medium supplies inorganic salts, carbon source, vitamins, and amino acids. Growth regulators, mainly auxins and cytokinins, control callus formation, root induction, and shoot regeneration. The cultures are maintained in controlled environment chambers and handled inside laminar airflow cabinets, which supply sterile filtered air. For CUET-PG, remember, laminar airflow cabinet is used to maintain aseptic condition, not for sterilizing instruments. That is done by autoclaves. Now, we move to the next major topic, cryogenic preservation or cryopreservation. This technique is primarily concerned with the long-term conservation of plant genetic resources. Cryopreservation is defined as the storage of biological materials at extremely low temperatures, typically minus 196 degrees Celsius, which is the temperature of liquid nitrogen. At such low temperatures, all metabolic and enzymatic activities practically stop, meaning that tissues can be stored for years or even decades without aging or genetic change. The main purpose are germplasm conservation, maintaining genetic stability, and protecting rare or endangered plant species. For CUET PG, the temperature value of liquid nitrogen is very frequently asked, minus 196 degrees Celsius. Materials preserved by cryopreservation. Various types of plant materials can be preserved using cryopreservation. These include meristems, embryos, callus tissues, seeds, pollen grains, and cell suspension cultures. Meristems are especially popular because they are small, actively dividing, and often free from virus, making them ideal for regeneration after thawing. Steps in cryopreservation. The procedure of cryopreservation involves several sequential steps which you should memorize carefully for CUET-PG. First, healthy and actively growing materials is selected. Then, pre-treatment is given, often involving osmotic dehydration or cold acclimatization. Next, cryoprotectants such as DMSO, Grisolol, or sucrose are applied to prevent damage caused by ice crystal formation. The material is then frozen, usually rapid, and stored in liquid nitrogen. When needed, the sample is thawed carefully and transformed to fresh medium for recovery and regeneration. CUET often asks which chemical act as a cryoprotectant. Remember DMSO as a standard example. Advantages of cryopreservation. Cryopreservation offers many advantages. It allows extremely long-term storage with minimal space requirement. There is almost no genetic change because metabolic processes are altered. Maintenance costs are low compared to field gene bank. The materials is protected by pests, pathogens, and environmental disasters, and biodiversity can be conserved efficiently. Therefore, cryopreservation forms the backbone of many modern germplasm conservation programs. We now move to developmental pathways, specifically organogenetic differentiation. Organogenesis refers to the formation of organs such as roots, shoots, and leaves from cultured tissues. This process is strongly influenced by the balance of plant hormones in the medium. Typically, a high cytokinin to auxin ratio promotes shoot formation, whereas a high auxin to cytokinin ratio encourage root formation. Organogenesis is widely used in micropropagation and regeneration protocols. There are two major types of organogenesis. First, direct organogenesis. Organs develop directly from the explant tissue without passing through a callus stage, which reduces the chance of genetic variation. Second, indirect organogenesis. A callus is first formed and then differentiates into organs. For CUET-PG, remember, presence of callus indicates indirect organogenesis. After understanding organogenesis differentiation, we now entered a very important session called types of plant culture. This is one of the most commonly tested areas in CUET PG because different culture types are associated with different applications such as embryo rescue, metabolic protection, germplasm conservation, somatic hybridization, and virus elimination. In plant biotechnology, depending on the aim of the experiment, whether we want to regenerate a plant, preserve genetic material, study development, or produce chemicals, we select a particular type of in vitro culture. So, as we go through each type, try to mentally connect techniques with application because CUET often frames questions in that way. This slide gives a bird's eye view of the six major categories of plant tissue culture: seed culture, embryo culture, callus culture, organ culture, cell suspension culture, and protoplast culture. All of these techniques are ultimately based on the same fundamental principle, totipotency, but differ in the kind of explant used and the experimental objective. Seed and embryo cultures are most related to developmental studies and breeding programs, whereas callus and cell suspension cultures are extensively used for regeneration and secondary metabolic production. Protoplast culture is unique because it involves removing the cell wall and is especially important in somatic hybridization and genetic engineering. CUET PG sometimes asks direct clarifications or classifications type questions such as which of the following is not a type of plant tissue culture. So you should be very familiar with the list. Seed culture refers to the in vitro germination of seeds under sterile laboratory conditions. In nature, seeds germinate in soil, but in the laboratory, they are placed on agar-based nutrient medium after surface sterilization. This allows researchers to observe germination processes without interference from microorganisms. Seed culture is particularly used for germplasm conservation, for studying the physiology of germination, for eliminating seed-borne pathogens, and for propagating rare or endangered plant species whose seeds may not survive well in natural conditions. In some orchids and medicinal plants, seeds are extremely tiny and lack sufficient food reserves. So, in vitro culture becomes essential for successful germination. For CUET-PG, remember that seed culture deals with whole seeds, whereas embryo culture involves excites embryos. Embryo culture involves the removal of an embryo from a seed and growing it artificially on nutrient medium. This technique is widely used in plant breeding to overcome problems such as seed dormancy or embryo abortion in white crosses, where two distinctly related species are hybridized. In such crosses, the embryo may start developing but fails to mature inside the seed because the endosperm does not support it adequately. By accessing the young embryo and culturing in vitro, scientists can rescue the hybrid, hence the term embryo rescue. Embryo culture is also used to speed up breeding cycles and to study the stages of embryogenesis under controlled conditions. CUET-PG frequently includes questions like embryo rescue is achieved by which technique? Correct answer is embryo culture. In callus culture, the explant such as a leaf or stem segment gives rise to a mass of unorganized, actively dividing cells called callus. Callus formation is usually induced by supplying appropriate concentrations of auxin and cytokinins in the culture medium. High auxin levels generally favor callus induction. Throughout callus tissue does not show organized structure. It is extremely valuable because it can later be induced to regenerate shoots or roots, making it a key intermediate stage in many tissue culture protocols. Callus culture are also important in genetic transformation experiments because Transformed cells can be selected at the Callus stage and later regenerated into whole plants. In addition, Callus or derived cell suspension cultures are used for the production of secondary metabolites such as alkaloids, pigments, and pharmaceuticals. For CUET-PG, remember the somoclonal variation. Genetic variation arises during tissue culture, is most commonly associated with Callus culture. Organ culture refers to the in vitro maintenance and growth of isolated plant organs such as roots, shoots, leaves, or even flowers. Unlike callus culture, whether the tissue is unorganized, organ culture preserves the natural organization of the structure being cultured. This technique is frequently used in developmental biology studies to understand how specific organs grow and respond to hormones or environmental factors. It is also employed in virus elimination programs, especially through shoot tip or meristem based organ cultures, because viruses are often absent or present in very low concentration in these regions. In CUET-PG, organ culture is usually tested through its applications, particularly developmental studies and disease elimination. In cell suspension culture, small cultures of cells or single cells are grown in a liquid nutrient medium, usually under constant shaking to keep them eventually dispersed and well oxygenated. Such cultures are generally derived from fibrillar callus tissue that easily breaks apart in liquid medium. Cell suspension cultures are extremely important for biochemical and physiological studies because they provide a relatively uniform population of cells that can be sampled easily. From an industry point of view, they are widely used for large-scale production of secondary metabolites in bioreactors, similar to fermentation tanks used in microbiology. For CUET-PG, associated cell suspension culture with metabolic production and scale-up in bioreactor. A protoplast is defined as plant cell whose cell wall has become or been completely removed, leaving only the plasma membrane surrounding the cytoplasm. Protoplasts are usually obtained by treating plant tissues with cell wall degrading enzymes such as cellulose and pectinase. Once isolated, protoplasts can be cultured in suitable media where they regenerate their cell wall, divide, and eventually form callus, from which whole plant can be regenerated. Protoplast culture is especially important in somatic hybridization, where protoplasts from two different species are fused to form hybrid cells that might not be achievable by sexual crossing. CUET-PG often asks enzyme-related MCQs here. Remember cellulose and pectinase. We now move to one of the most commercially important applications of plant biotechnology, micropropagation. Micropropagation refers to the rapid multiplication of plants under in vitro conditions and forms the basics of large-scale production of planting material for agriculture, horticulture, and forestry. This technique has revolutionized the cultivation of crops such as banana, sugarcane, orchids, potato, and many ornamental plants. Micropropagation is defined as the rapid in vitro clonal multiplication of plants using tissue culture techniques. The word clonal is very important. It means that all the plants produced are genetically identical to the parent plant. This genetic uniformity is desirable when we want to multiply alike varieties with no superior traits. For CUET-PG, the phrase rapid clonal multiplication is frequently used in MCQs as a clue pointing to Micropropagation. The process of micropropagation is usually divided into 5 stages. First is selection of the explant, choosing healthy, disease-free source material. Second is establishment of the culture, whether the explant is derived and placed on a medium. Third is shooting multiplication, achieved by manipulating cytokinin level. Fourth is rooting of the shoots, generally promoted by auxins. Finally comes acclimatation or hardening, where the plantlets are gradually transferred from laboratory conditions to soil and natural environment. CUET PG commonly asks sequencing questions based on these stages, so memorize this order carefully. Axillary bud involves stimulating the growth of dormant buds located in the leaf axils. Because of these buds are pre-existing meristems, the plants produce are usually genetically stable with very low chance of somochronal variation. For this reason, axillary bud is preferred method for commercial micropropagation. It is widely used in crops like banana and many ornamentals. In CUET-PG, if the question mentions commercially preferred micropropagation techniques with low variation, the answer is axillary bud. Meristem culture involves isolating and culturing the apical meristem, the tiny region of actively dividing cells at the shoot tip. Viruses usually fails to invade these rapid dividing cells, so meristem culture is extensively used to produce virus-free plants. This is especially important for vegetatively propagated crops like potato, banana, and citrus, where viral infections can be accumulated over generations. CUET-PG often asks application-based questions such as which technique is used to eliminate viruses from plant stock. The correct answer is meristem culture. Shoot tip culture is closely related to meristem culture, but involves a slightly larger piece of tissue that includes the meristem along with a few surrounding leaf permodia. Because the tissue pieces is larger, survival rates may be higher than in meristem culture. Although the efficiency of virus elimination might be slightly lower, shoot tip culture is used both for disease elimination and for rapid multiplication of allied plants. In CUET-PG, if a question distinguishes between meristem culture and shoot tip culture, remember that the key difference lies in the size of the explant. Bud culture refers to the in vitro cultivation of axillary or terminal buds. It is used primarily for clonal propagation and for conserving germplasm of valuable genotypes. Because buds contain organized meristematic tissue, this method is relatively reliable and produces genetically uniform plants. Bud culture is sometimes considered under the broader umbrella of micropropagation techniques. Now we move to the two major regeneration pathways in plant tissue culture: organogenesis and somatic embryogenesis. In organogenesis, roots and shoots develop separately from cultured tissue, usually in response to changes in hormone concentrations. In contrast, somatic embryogenesis involves the formation of complete embryo-like structures from somatic cells, which then germinate to form whole plants, much like zygotic embryo. Organogenesis is generally considered less complex, whereas somatic embryogenesis is highly organized and mimics normal embryonic development.
CUET PG frequently includes comparison between questions based on these two processes. Somatic embryogenesis is the process by which embryos are formed from non-reproductive somatic cells rather than from fertilized eggs. These somatic embryos pass through characteristic developmental stages listed in the screen. This technique is very important and powerful because a single explant can produce large numbers of embryos, making it useful for mass propagation and synthetic seed production. For CUET-PG, the sequence of stages is very important. Memorize them in order. Applications of somatic embryogenesis. Somatic embryogenesis has several important applications. It is used to produce synthetic seeds, where somatic embryos are encapsulated in protective coating for storage and sowing. It supports clonal propagation of elite plants. It is valuable in genetic transformation experiments because embryos can regenerate efficiently into whole plants. It also contributes to germplasm conservation. CUET-PG may ask which regeneration pathway is associated with synthetic seed. The answer is somatic embryogenesis. Finally, we analyze the strengths and limitations of micropropagation. Micropropagation offers several advantages, including rapid multiplication of plants throughout the year, independent of seasonal constraints. It allows production of disease-free stock and genetically uniform clones. It aids in the conservation of rare and endangered species and often results in high survival rates once plantlets are properly hardened. Because of these benefits, micropropagation has become a cornerstone of modern horticulture. Despite its many benefits, micropropagation also has limitations. Setting up tissue culture laboratories is expensive and requires skilled personnel. There is always a risk of contamination by microbes. Somoclonal variation may arise, particularly when regeneration occurs through callus culture. Finally, transferring delicate in vitro grown plants to natural soil conditions during acclimatization is challenging and requires careful management. CUET-PG often includes questions on these drawbacks. To summarize this unit, plant biotechnology relies heavily on tissue culture techniques based on totipotency. Different in vitro culture types serves different purposes. Micropropagation is widely used for commercially, and cryopreservation enables long-term storage of germplasm. Organogenesis and somatic embryogenesis represent two distinct regeneration pathways. This unit forms the conceptual backbone for later topics such as genetic transformation. Before we finish, students, please check the description box below this video. There you will be find the registration link for Unit 1 PDF notes, previous year based questions, most expected questions for CUET PG 2026, personalized study schedule, and revision material. These resources are meant to support your preparation along with the lectures. Watching videos is only first step. Regular practice and revision are equally important. If you need any academic support, the contact details are also provided. Do use them whenever you feel stuck in any topic. And with this, we conclude today's lecture on Unit 1 Plant Biotechnology for CUET PG 2026. Revise today's topics carefully, especially totipotency, cryopreservation, types of culture, micropropagation stages, and organogenesis versus embryogenesis. If you found this video session helpfully, Do subscribe to the channel and share it with your friends preparing for CUET PG. Thank you for watching. Keep studying consistently and I will see you in the next class.